Hello YouTube, Joseph here with Prep for Step. I'm back with another video. Many of you requested for me to make a video about the JAK-STAT signaling pathway, so here it goes. The JAK-STAT signaling pathway is obviously a signaling pathway. Now, in order to understand it better, let's understand what the actual components are and what do they mean, okay? So the JAK-STAT signaling pathway has three main components. One is the receptor on the cell surface of some particular types of cells, right? So it's on the cell surface. All right, now this cell surface receptor binds to specific signals or specific ligands. Now what I like to do is I like to call these ligands, because only certain ligands bind to this cell surface receptor. In order to memorize them, I have something called TINs and TERs, right? This is TINs and this is TERs. Now the TINs would be a few different hormones such as prolactin, erythropoietin. Another one would be, let's see, thrombopoietin. And another one would be leptin. The TERs refers to interferons and interleukins. Okay, so this is how I remember this. There's also another one that you can add if you want to. It's the basically GM-CSF, which is the granulocyte macrophage colony stimulating factor, which is secreted from certain cells such as macrophages, and it stimulates the growth of white blood cells and monocytes, which then turn into macrophages. So these guys basically are the ligands that attach to this cell surface receptor and activate this particular pathway. Okay, so the second component of the JAK-STAT signaling pathway is called JAK. Now, what is JAK? Well, JAK stands for Janus kinase. Okay, Janus means it's a Greek word, I believe, for God of transition or God of the beginning. Okay, and kinase is basically any type of enzyme that uses ATP to phosphorylate other proteins. And why do we phosphorylate other proteins anyway? Well, phosphorylation of some proteins activates them. Phosphorylation of some other proteins deactivates them. So it's pretty much turning on and off different proteins. So the JAK, that's basically what the JAK stands for. The third component is going to be the STAT pathway, or the STAT component of the signaling pathway. And what does the STAT stand for? Well, basically, it stands for the signal transducer and activator of transcription. I'm going to horribly butcher this out, but anyway. So, STAT, signal transducer, and activator of transcription. So what do you think is going to happen? Well, let's say we set a receptor is there. There are certain ligands that bind to this receptor. This receptor is then going to activate some kind of JAK. JAK, by any chance, is going to do what? It's going to phosphorylate something. And that phosphorylated something, most likely it's going to be a STAT. And this STAT is going to transduce that signal and activate the transcription, which in order to do that, it's going to have to enter the nucleus. Okay, so let's summarize all that we've talked about thus far in this particular image. It's a very good image. It shows everything from start to finish. Okay, so the first step, we said that the ligand, in this case I chose erythropoietin. You can choose GM-CSF. All the interleukins and interferons, the TINs and TERs that we talked about, can basically be this instead of erythropoietin. This ligand is going to bind to the receptor. As you can see here, it's bound. Once it binds, it's going to activate JAK. Now, JAK, we said, is a kinase molecule. Kinase meaning it's going to use ATP to phosphorylate something, to put a phosphate on something. In this case, it's actually phosphorylating tyrosine residues on itself, which is why it's called a autophosphorylation of tyrosine residues. So it's going to phosphorylate, adding these little phosphor groups on tyrosine on itself, which activates itself. Now, once this happens, these STAT molecules floating around in the cytoplasm, come and attach to these phosphorylated tyrosine residues. So you can see STAT is now attached. Once that happens, JAK now is also going to phosphorylate. It's going to use, again, another ATP to phosphorylate the STAT proteins. The STAT protein then dimerizes. Dimerizes basically turns into a unit that has two compartments or two components, which both of them being STAT. So this is a dimer of STAT protein. Now this STAT dimer is going to actually enter the nucleus, and this is our nucleus. It's going to enter the nucleus, and it's going to bind to certain specific regions on the DNA, and it's going to promote translation of that particular sequence, or translation of a particular sequence, and it's going to promote that. So it's going to bind to promoter regions on the DNA. The result being some type of protein, eventually some type of mRNA is getting transcribed, and then the mRNA is going to be translated into certain type of proteins, which then they go and take their effect. Now, for those of you that are going to take step 1, USMLE step 1, this is pretty important, okay? This is the clinical significance of this particular pathway. Now, if by any chance this pathway becomes mutated, either through inherited or some type of acquired genetic defects, if there's any change in this particular pathway, it could lead to some type of immune deficiency syndrome or some type of cancers. Now, there are 4 different types of JAK proteins, okay? There's JAK kinase 1, 2, 3, and there's a tyrosine kinase 2, or TYK2, which is also part of this family. The most important of these being the Janus or Janus kinase 2, okay? Now, what's important about this? Well, the Janus or Janus kinase 2, or the JAK2, is mutated in a lot of the chronic myeloproliferative disorders, okay? Now, in the case of polycythemia vera, what's the problem? The JAK2 is, remember, for polycythemia to get it, you have to have erythropoietin, right? You need to produce red blood cells. You need that hormone, which is erythropoietin. In this case, the erythropoietin doesn't even need to bind to the JAK2. Okay, because the JAK2 is already mutated. If it's mutated, it's activating, it's auto-activating itself more and more and more. So the patients get a lot of red blood cells, but at the same time, their erythropoietin is not that high. So in 100% of the patients with polycythemia vera, this particular enzyme is mutated. It is also mutated in 30 to 50% of patients with essential thrombocytosis, pretty much producing too much platelets, and myelofibrosis, or fibrosis of the bone marrow. The only myeloproliferative disorder that does not have a mutation in the JAK2 pathway is CML. Okay, so I hope this helps you guys. Good luck, and we'll see you next time. Please do subscribe. I'm going to be uploading a lot more videos from now on. Thank you very much, and see you next time.